Okay, good afternoon. My name is Maria Orio. I'm the head of regulatory affairs at Viralgen. And first, I would like to thank you, Anna, and the CDMB1 Foundation team for this invitation. I would have loved to be here on site with you and meet you in person. Unfortunately, it wasn't possible for me either, like Cesar. But in any case, I'm really happy to be here and really looking forward to talking to you today about the translational challenges of advanced therapies from a regulatory perspective and based on our experience here at Viralgen. So today, the agenda of the topics we will be talking about will be what the steps are from new medicine to market, so from bench to commercialization, and then we will zoom in a little bit to see what the details are on the steps that are needed to be taken from preclinical phase to market. After that, we will briefly talk about the challenges that we are facing in gene therapy manufacturing, and at the end of the session or even after, there would be a Q&A session where I will be more than happy to answer any question you may have. So in this slide, we see the steps that need to be taken, or the pathway from bench to market. So these are the steps that we need to follow in order to be able to go from early development of a new drug, going through preclinical phases, then moving through clinical phase, and finally putting the product into the market. So at the very earliest stages, at the drug discovery and development, it's where we would perform all the screening and discovery of new active substances. Then after this, we would move to preclinical phases. This is where the material is used in animal models and where we'll be getting data to see the pharmacological efficacy, pharmacokinetics, and toxicity studies. And after the preclinical phase is completed and this preclinical data is successfully generated, we would move to clinical phase, where we will start using or testing this material of this new drug in humans. In these clinical phases, here in this representation, we can see the three different clinical phases of a classical or a traditional approach of a clinical trial, is where we would confirm in human safety and efficacy. And after this, clinical phase is completed, and again, the clinical data is successfully generated, we would move to commercial phase, where we would file all the data that we have generated so far from preclinical phase and clinical phase, and some other CMC data generated in order to be able to put the product in the market. And once the product is marketed, of course, there will be a life cycle management where there will be a follow-up and continuous monitoring of the safety of the product from a pharmacovigilance perspective. And how does Viralgen support this journey? Viralgen's mission is to support clients, our clients, and therefore the sponsors who are developing new drugs from the development to commercial manufacture of their gene therapy drugs, in order to ensure that they obtain material of the highest quality at the right time and at the right scale to meet the needs of the patients worldwide. So this is our aspiration, this is our true north, and this is what inspires us, because we really want to help these patients and these sponsors to get this material for the treatments. And if having in mind this mission and how Viralgen would like to support the sponsors in this journey, if we go back to the previous slide, then we see that Viralgen support this journey by producing multiple scales and grades of manufacturers. So for instance, if we have a look now at the left-hand side of the slide, at the very early stage of the development, we at Viralgen produce 2-liter batches for the feasibility studies. And after that, we produce toxicology batches of 50-liter scales in order to be able for this material to be used in the preclinical studies in the animal model. As we said before, once the preclinical data is generated, then we would move to clinical phase. In this case, we would be producing material at 250 and 500 liter scale under GMP conditions, which is what Cesar mentioned earlier. These are the conditions that need to be followed in order to be able to put material into humans. So Viralgen can support this transition from lab to clinical supply. And of course, we are also able to produce material at clinical scale in order to be able to do the different tests and exercises for the marketing authorization approval, or BLA. And we are able to produce this commercial material at different scales, such as 250, 500, and 2,000 liter scale. If we slightly zoom in this journey or this pathway, we can see what the steps would be from preclinical phase to market. So here, it's where we would be today with this program. We are at the preclinical phase. We are generating this data, and once we have generated this data, the next step would be to file an IND or IMPD in order to get the approval for a clinical trial. This would need to be done via a clinical trial application in the country where we would like to run the clinical trial. The clinical trial would be a cycle where we will do the assessment, definition of further trials, and the study of specific population groups. There's many documentation that needs to be submitted. Today we won't go in that depth because it's not the scope of this session, but I think it's important to highlight that we need to ensure, of course, the safety of the patient at all time. Once we have generated all the clinical data, we would be able to go for the submission for approval, the marketing authorization application or biologic license applications in the US. And once we get the marketing authorization approval, it's when we will go into the life cycle management, and we will have to do the pharmacovigilance. We would enter into the pharmacovigilance cycle where we will do the continuous monitoring of the drug. So what we have seen so far is the traditional approach. This is what the guidelines say and what the agencies are expecting to see. However, today, and given the complexity and characteristics of gene therapy, we are facing many challenges, because in gene therapy, 1 + 1, it's not always 2, and basically I think it's never 2, actually. So how do we sort out these problems or these challenges that we have, and what are these challenges actually? So what we have seen so far is that the regulators are evolving. They are changing and intensifying their requirements. They are becoming stricter. The regulation is very generic. There's nothing really specific for gene therapy, and this is a huge challenge for us because there's no clear pathway for us to follow. So basically, we are learning by doing every time. And an example of this would be that I would say 1.5 years ago, it was widely accepted by many agencies to use the ITR qPCR as a titer method. However, currently the agencies are strongly recommending to move to transgene ddPCR as a titer method of choice. The problem or the challenge here is that in order to be able to do this, we need an analytical development behind. This means, of course, resources and time. Again, a challenge. As Cesar mentioned before, one of the characteristics or challenges of these therapies is the small patient population. This means that we will require only a limited number of batches for these therapies. The problem is that with this very limited number of batches, the knowledge of the product is very limited as well. And in order to be able to set acceptance criteria, or at least to know how the product behaves, we really need to produce material. So this is again a challenge, because the less material we have, the less knowledge we will have about the problem. We need to find a creative solution, of course, for this. So this is something Viralgen is working on, and that's basically the reason why Viralgen is doing what we do, producing these kind of therapies at multiple scales and using a platform. This limited amount of knowledge and this challenge in terms of the ability to generate sufficient data, it's also a challenge in order to be able to develop acceptance criteria for certain critical quality attributes and control strategies. So I can give you another example here based on our experience is that maybe less than 2 years ago, it was accepted by the different agencies to go with report results for many of the quality attributes, especially for residual impurities, if we were talking about early phases of clinical trials. However, we are getting more and more the questions to get or to set acceptance criteria, even for early clinical phases. And again, this is a challenge because of the limited knowledge of the process. How does, again, Viralgen support this journey? The challenges are there. This is something we cannot change. The industry is evolving. Hopefully in a few years, there will be more knowledge about the process and the product. But today, this is where we are, and we really need to find a solution in order to be able to put this therapy into the patients. So what Viralgen is doing and the way we support the sponsors in this journey is basically gaining knowledge based on the continuous improvement by receiving feedback from the different regulatory agencies. So for instance, there are currently 12 clinical trials approved worldwide with material produced at Viralgen. And out of these 12 regulatory submissions, we have responded to over 20 lists of questions. This means data, this means knowledge. So every time there is a submission, every time we have an opportunity to interact with any agency, we are improving and increasing our knowledge. This is pure gold for us. And the same for the data. So the more material we produce, the more sponsors we work with, the more data we will be able to generate. And based on the platform power, we will be able to leverage the historical data to support other products. Even if it's not the same product, we will be able to cluster data per serotype or per platform, and we will be able to see what we can do to support these different packages. And then if we look a bit forward and put an eye on the next step for the marketing authorization stage, the traditional approach or what the process validation guidelines say is that a full process characterization and 3 successful PPQ runs are required. However, if we have in mind, or if we're talking about rare and ultra-rare diseases with very small patient population, single dose, limited number of batches, does it really make sense to produce 3 consecutive PPQ batches at a commercial scale? With this, we may have material to treat the entire population for several generations. So we really need to have a conversation with the agencies and maybe challenge this requirement and see what the options are for us. So finally, I would like to close this session to say that we are here to bring hope to the patients, because what we have seen so far in our experience is that the agencies are eager to support gene therapies. They're open to see new approaches, and they are really encouraging the sponsors to be creative, not only in terms of the clinical trial design, but also in terms of CMC. The agencies would really like to have a close and transparent relationship with the sponsors. And this is definitely an open door for us. And I would say that we really need to work together to make the most of this opportunity. Thanks so much for your time.